very happy to introduce today Professor Peter Hong. He is the Deputy Director for the Center for Brain Research and is a member of the Management Directorate of Brain Research New Zealand, which is one of the national centers or centers of research excellence. So Peter finished his PhD at Auckland and then went on to do postdoctoral studies at Auckland and then the Kresge Hearing Research Institute at the University of Michigan. He now leads the Audiology Neuroscience Group at FMHS, which has a research focus on auditory neurobiology, particularly inner ear homeostasis, cochlear neurodevelopment, the influence of noise and aging on hearing, and new diagnostic techniques such as MRI for ear disease. So Peter has held many senior management positions at the university, and he works closely with community hearing disability organizations and NGOs in the audiology profession to enhance services for people with hearing impairments.
Thanks, Renan. Thank you very much, and thanks for the opportunity to come and talk to you today. I thought I'd just give you a bit of an idea of the sort of outline I want to talk about, because what I'd like to try and do is just really give you a bit of an overview of some of the things that we're doing, but with a particular focus on some of the work that's going on in the sort of therapeutic approaches to try and treat ear disease in particular, and some of the issues that we have for trying to translate some of this research into clinical practice. And we thought that maybe towards the end we could just talk about some of the ways that perhaps there could be some collaborations with engineering. We've already started one with, this year with one of the fourth year projects, trying to make a small coil for MRI imaging of small rodents, and there's some opportunities I think perhaps for looking at trying to get some collaborations going. So we'd be really delighted to talk about those and talk about them further. So the first thing I thought I'd do is just talk about hearing loss. This is a very awkward room, so if I go up here I can't see what I'm talking about. Or it's
better off just wandering in the middle.
Okay, so look, I'll just... Oh, no? Oh, okay. I can't wander because apparently I'm being filmed. So the thing is just to give you a bit of a feel for why we think there is a problem, and there is a problem to study. The prevalence or incidence of hearing loss in deafness in particular, very severe conditions in babies, is very high. So in New Zealand it's between 1 and 3 per 1,000 babies are born with a significant hearing problem, and if you go into some of the developing countries it can be up to about 7 or more per 1,000. So it has quite a high incidence. And then that starts to increase as you look in, for example, intensive care, particularly around asphyxia at birth, and jaundice and so forth can lead to quite significant hearing problems. And then of course for those of us who are sort of heading into this bit, there's a very, very high prevalence as you get into as you age, and this is one of the reasons why this is becoming even more of an issue, because the projections, if we think about somewhere between 10 and 70% of the New Zealand population has quite some degree of hearing disability, there's a belief that that's going to increase to around about 27% in the next 30 years or so. So you young ones, you're in trouble. So I thought just first of all just think about the types of hearing loss that we're talking about, because just to give you a bit of a flavour for it, so that we can divide the hearing loss into 3 types really. One which is what affects the middle part of the ear, which is the small bones that transfer the sound across into the inner ear where the transduction occurs. And we often refer to that as conductive hearing loss. So it's because there's a change in the transmission properties of the middle ear, and so the sound effectively doesn't get in. So it's really just like turning the volume down. It has a very limited effect on frequency analysis by the inner ear. The predominant one, which I'm going to talk about more later on, is what we call sensorineural hearing loss, and that's associated with this is my finger over here. We should have a way of being able to project the finger over to the other side. So this is actually in the inner ear. So there's the cochlear and the vestibular apparatus. So the often diseases that affect the inner ear cause both hearing and balance problems as well. And then we sort of think of central problems, and we're getting to know far more about this now as people are recognising there's a lot of central auditory processing difficulties. So people have problems in processing of sound, which is often associated with abnormalities in the auditory pathways. And this seems to be a lot more prevalent now as we're starting to get better tests for, and maybe it's also underpins things like some conditions like dyslexia, is starting to be seen to be associated with conditions like autism and so forth. So it's maybe just really demonstrating a sort of a global issue of processing, and particularly sensory processing in the brain. In terms of causes, there are congenital and perinatal factors. As I mentioned, asphyxia at birth can be quite a significant problem in causing hearing loss, particularly as associated with the brainstem and the cochlear. Genetic conditions, there are about, last count's about 40 genes now which are known to be related to deafness, and there's about 120 mutations, so there's quite a significant genetic impact on hearing. Infections such as middle ear infections, which cause that conductive hearing loss that I was talking about, so that's things like glue ear, which you might be familiar with. Trauma is another one. Metabolic conditions, these really relate to problems of homeostasis inside the inner ear. So as I'll show you a little bit later on, is the inner ear has a fluid system, and that is tightly controlled. Its ionic composition is tightly controlled, and there are conditions which lead to deafness as a consequence of changes in the ionic homeostasis. Ototoxic drugs, things like aminoglycoside antibiotics, so they cause deafness. You don't see that so much in New Zealand because we have other antibiotics, but you go into the developing world and developing countries where aminoglycoside antibiotics are heavily used, there's a very, very high incidence of profound deafness as a consequence. So it's essentially survive the condition and go deaf, or don't survive. So, and because these antibiotics are very, very cheap, they do have quite a significant effect. We've been working up in Fiji, and there they have a real problem in their neonatal units because they use aminoglycosides a lot, and they have a lot of deaf babies. So question of how to actually stop that. Noise exposure and age-related hearing loss, some of the work which we've done would suggest that about 25% of the burden of hearing loss and sensory neural hearing loss is due to noise exposure. So actually trying to stop the noise exposure, both domestic and industrial, would have a huge impact on reducing the burden and the amount of hearing loss in the community. The central pathways and central processing disorders is really interesting, and there are some particular conditions. There's a condition called auditory neuropathy. There's a newly discovered condition. I'm sure it's been there for a while, but it's amblyaudia, which is, so you would be used to amblyopia, but this is where the ears are actually offset. So one ear, you have a sort of a lazy ear, and that has quite significant effects on the ability to process binaural signals. And so there's a lot of interest in trying to retrain the ears using similar sorts of things to what you would do with amblyopia, of actually either reducing the activity on the good side or boosting the activity on the other side to improve it. Tinnitus, you may all be familiar with that, especially if you attend concerts and things. But that's actually becoming, it's really quite prevalent in the community. The other thing which is kind of interesting and is really starting to become quite well recognised now, and there's a lot of work going on, is the relationship between hearing loss and some of the other conditions. And some of these, like cardiovascular and diabetes, perhaps we can understand because they have similar issues around vascular disorders and so forth. But this is an interesting one, this relationship between dementia and the development of dementia and cognitive decline and hearing loss is starting to show there's a lot of evidence of quite a strong correlation, and there is some evidence that you're about 8 times, I think it's about 8-fold increase in risk of developing Alzheimer's. If there is a decline, if you've got cognitive decline, then chances of actually developing a more severe dementia is much, much higher if you have some sort of auditory processing disorder or hearing disability. And this may just relate to the fact that, like some other sensory disorders, that there's a relationship between the actual cognitive burden of trying to deal with a hearing loss as well as having this generalized decline in cognition. So just to talk about, just now moving on from that and just thinking about some of the things that we're doing, and just to give you a bit of an outline of the broader group, which Vinod just outlined very briefly. So across Tamaki and Grafton, we have a number of groups that are working in the sort of general area of audiology and hearing sciences, and I realise Fabiana's here. I should have included you, Fabiana, in this, but terribly sorry.
I'll get you later.
You'll get me later, I know. and she will. I mean, I can assure you. She's not just joking. So there are a number of groups that are working in this, and we're all sort of interconnected in some way, but I'm going to talk mostly about some of the work which we're doing at Grafton and the Auditory Neuroscience Group. But there are a lot of clinical studies going on into age-related hearing loss and hearing technology, and you might like to get Grant to come and talk to you about that, because he may have done that before, but there's some very interesting work he's doing in the area of tinnitus. And we've also got quite a big effort going on in the sort of population health area, particularly around hearing loss prevention and health systems. And looking here, it's mostly around noise exposure. And there's some quite innovative things being done. There's programs of things like the Dangerous Decibels program, which is a really interactive school education program to try and teach kids to understand and to support their hearing better, and to look at recognising the problems of noise exposure. And we've actually moved that now into industry and developing a program to try and educate people in the workforce about hearing loss and hearing prevention, hearing loss prevention using much more interactive programs. So just drilling down now to the Neuroscience Group, and just sort of where I want to sort of head with this is to talk to you about some of the work we're doing. And the sort of key projects that we have underway and have been doing for a while is there's a program of work which we've got looking at the development of the neural innovation, and particularly how the auditory nerve connects with the sensory cell and develops its synaptic connections, and also projects the other way into the cochlear, into the brainstem. And because that's highly vulnerable during during development and at birth, because that's where the asphyxia problems seem to occur, is actually particularly in the connections into the brainstem. And so we're just trying to understand much more about how that works. And we've got an animal model of asphyxia that we're looking at, the sort of neural innovation associated with asphyxia. Probably the majority of work we've done is looking at, with animal models, of how does the cochlea, the inner ear, degenerate under a number of different conditions, and I'll talk about some of those. And this has sort of led on to how can we protect the inner ear, or try to prevent the degeneration that leads to the hearing loss after such things as noise exposure, and particularly with age-related hearing loss and ototoxic drugs. So we have a number of programs looking at mechanisms of action of aminoglycosides. How does age lead to, how does the degeneration occur with age, and the interaction with that and noise exposure. I want to talk much more today about some of the imaging work which we're doing to try and understand more about inner ear disease processes, and we're starting to develop now with Brain Research New Zealand, the new core, of some work going on relationship between hearing loss and cognitive decline, looking at animal models of that. So what I'd like to do is just focus on these two and talk a bit more about these. And this is perhaps where there might be some interesting interactions that we could develop with engineering. Please stop me if there's questions you want to ask, or tell me to go away or something. Now, the thing that, when we talk about sensorineural hearing loss, is that the majority of the sensorineural hearing loss occurs because of changes inside the cochlea of the inner ear. And so the cochlea is the spiral structure. It's about, the human one's about the size of your little fingernail, something about the order of just from the first joint there. So it's about 3 or 4 millimeters in it's a cone, 3 or 4 millimeters in diameter, and at the base, and about 5 or 6 high. So it's and it has the spiral structure. So if you just pick up any spiral shell off the beach, that's exactly what the cochlea looks like. It's just a fluid chamber that wraps around a central column, which contains all the nerve fibers, as you see here. And it's attached to the vestibular organs, the semicircular canal, and the vestibule here, which contain the balance organs, or the gravity receptors and accelerometers, effectively, that and they have the same nerve that passes back to the brain, connected with the auditory nerve. So if you go back to this, if you just cut an axial section down this in the ear, this is a human temporal bone, and all this white area here is the middle ear. And you can see the stapes there, the last bone of the ossicular chain, That goes into the vestibule, which contains the otolithic organs, the sacral and the utricle. This is one of the semicircular canals which would be wrapping around like that. And this over here is the cochlear, and you can see that you can imagine that spiral structure with the axial section here. And this is the nerve fibers coming out and going into the internal auditory canal, and the brainstem is just about here. There's a couple of things just to point out here. One is that the relationship between the middle ear and your brainstem is very small. I mean, that's just a small space. And there's a small ligament which attaches the bony footplate of the stapes here. So there's a lot of opportunity for passage of infection between the two compartments, between the middle ear and the brain, and in the opposite direction. So this is one of the problems, and going into here, this is one of the problems, for example, with meningitis, that there are infections in the meninges which actually then end up inside the cochlear, which is why deafness is such a huge consequence of bacterial meningitis. And why also it's really important to limit infections inside the middle ear because of the potential for them to form brain abscesses or to go further. And especially if you've got corrosive enzymes which will then destroy the bone, it eats away and can lead to serious brain infections. So moving on from the gloom, thinking about the cochlear here, so what this has got the nerve fibers coming in here, and the sensory organ is out here on the periphery of the spiral. Now there are two fluid systems inside the inner ear. So if you just think of it, if you actually go back to this one, you might be able to make out a small membrane here. That's the inner tube or inner fluid space, and that's what's shown here in blue with inside the vestibular organs and into the cochlear. And so what's in red is what's filled with a fluid called perilymph, and that's just like any extracellular fluid, very high in sodium. And the blue one is filled with a fluid called endolymph, which is very much like intracellular fluid. It has a very, very high potassium concentration, and inside the cochlear it has a very high positive electrical potential, about 100 millivolts compared with perilymph. And that electrical potential sits at the top of the sensory cells, and it provides the driving force for the input of ions, particularly potassium ions, into the hair cells during transduction. So it's a very, very highly metabolic, the tissue here, very metabolic to regulate those ionic composition. So if you take away all the bone, and you'll see this is the organ of corti, this is the sensory organ here, or part of it. This is actually from a guinea pig. And you can see each of these little white tufts here are the sensory bundles of individual hair cells. So there's around about 12,500 hair cells in a human cochlear, and it makes up about 2.5 turns. And this is what's called the organ of corti. This is one with the sensory cells in it, and you can see there are two types of sensory cell. There's these ones called the outer hair cells, and another group called the inner hair cells. The outer hair cells are motor cells. So they actually express a motor protein in their lateral walls, and when they're stimulated, they contract and generate a force that then increases the motion of the basilar membrane, the motion of this in response to sound, and it drives the input to the inner hair cell, which is the main sensory input to the central nervous system. The outer hair cells have very, very few afferent fibers. There's a very small innervation from those fibers to the central nervous system. It's part of a gain control rather than actually sending any acoustic information to the central nervous system. The main input is from the inner hair cell, which has about 90 to 95% of the neurons. These neurons having different properties, which is what we've been studying a bit. And each of the hair cells has a bundle of stereocilia, or little sensory hairs that project. And if you can see a little bridge at the top of these, that's the transduction channel. That's the site of transduction. When these stereocilia move backwards and forwards with sound, it pulls on a membrane, a mechanically linked channel, mechanically gated ion channel, and this is all surrounded by potassium in the endolymph, and the potassium floods into the cell to cause the transduction, being driven by that very high positive voltage. So nearly all of the hearing loss which occurs because of inner ear injury comes from either the destruction or damage to the stereocilia, to the sensory hairs. So this can occur with noise exposure, for example. They seem to become fused together or can actually collapse like this, perhaps. We don't think it's purely because of mechanical injury per se. It's there's a change in stiffness and they get they fall over because of changes in the ionic properties that lead to the stiffness of these stereocilia. The cells can be completely destroyed and the whole pattern can disappear, and the cells can disappear and the neurons can disappear as well. So there's a lot of different pathologies, and I don't want to go into the different types, but all of these cells are particularly vulnerable to injury. But the most important thing, and why we really are looking to study so much of this, is that these sensory cells are all terminally differentiated cells, so they do not regenerate. So once they're lost, that's it. You can't regenerate the sensory cells inside the inner ear. And in addition is that their neurons, their dendritic processes are dependent upon the trophic relationship between those cells. So when the cells disappear, then the neuronal components start to retract. And that's clearly really an issue for things like cochlear implants, because you want to maintain a good population of auditory nerve fibers. So there's a lot of work trying to look at how to maintain those neurons. So really, in order to treat hearing loss, we need different approaches to either to develop regeneration of the cells or to protect or repair them. And so there's been a lot of work going on looking at development of therapeutic approaches, a lot of work looking at pharmacological therapies. There's some really interesting work going on in stem cells, particularly around the neurons. Now there are populations of stem cells which people have managed to inject into the inner ear and redevelop, I suppose, in a way, repopulate the neuronal population. And it's really interesting. These neurons, these stem cells enter into the inner ear. They form connections. They will form dendritic process, but also then send an axon all the way to the cochlear nucleus, and will innervate similar regions to where which would be expected according to their sort of frequency representation. So there's some really exciting work going on in this area. There's also some really interesting work going on in trying to turn on the supporting cells, which are the actual progenitor cells of the sensory cells, to turn on supporting cells to make new hair cells. And that's the genetic basis of that regeneration has been quite well worked out, and there's actually a clinical trial going on at the moment to see if to look at the regeneration of sensory cells and neurons, or particularly sensory cells. And prosthetic devices, you had a seminar here a year or so ago talking about cochlear implants, and that's it's a wonderful device. I'm not going to talk about that. What I'd like to do is really just focus now on the pharmacological side, because that's really where we've been working in that space. So just some very simply, one of the general view that's from a lot of the research which has been done, including a lot of the work we've done, is that a lot of conditions, particularly related to age and noise exposure, come as a consequence of generation of oxidative free radicals inside the cochlea, and the generation of an inflammatory response. And that seems to then perhaps have a bystander approach that then causes further degeneration and causes a lot of the injury. And this is just an example taken from a study by Henderson, where if you look at the green here, this is where there's evidence of free radical damage to membranes. So these areas here are the outer hair cell regions. This is in a control, and you can see here after noise exposure a high level of oxidative damage. And what's really interesting is that damage can occur for some time after the noise exposure. So it gives you the opportunity perhaps for a therapeutic window, because potentially if you can reduce that oxidative damage after, say, noise exposure, you may well then be able to recover or prevent the cellular degeneration and the hearing loss. So this has been the sort of target. This oxidative stress and inflammation have been some of the targets we've looked at. And cochlear inflammation, from a lot of different conditions, so ototoxic drugs, noise exposure, the introduction of a cochlear implant, so opening up the cochlea and inserting a cochlear implant causes an inflammatory response, which has a downstream effect leading to injury. And this is the sort of thing that you see, all this stuff here, you see the presence of immune cells surrounding the cavity here. This is all part of an inflammatory response generated by the inner ear, by the injury. And what's really very interesting is that the inner ear is to an extent immune protected. So it has a blood-labyrinth barrier. There's a barrier between the blood and the fluid systems, or all the tissue. And so it has its own immune system in a way. It has resident macrophages and resident immune cells, and these can be activated by injury. And it's thought that some of the injury might actually perpetuate a cascade of changes, which then leads to further injury, and is why even a small injury can actually cause quite a significant hearing loss over time. So, not to take too much notice of this, but this is just a whole series of drugs now or compounds which have been shown in animals to have protective effects. And nearly all of them are antioxidants, or they're inhibitors of stress response, or they're inhibitors of some of the terminal kinases which are associated with apoptotic processes or cell death. And over time, these ones here are ones which are in bold, which are now going into clinical trial. And this one here, the one in red here, the adenosine receptor agonist, this is an area which we've been working in substantially in way of to try to reduce oxidative stress and inflammation. And I'll just talk about it briefly. And for example, if you put in a compound which is an agonist of an adenosine receptor, and I won't worry about why we've gone this way, we can actually reduce the amount of hearing loss. So this is an audiogram you might be familiar with in an animal. This is taken by measuring the auditory brain response. This is the threshold of the normal prior to giving noise exposure. This is after the noise exposure, and if you treat these animals, we can bring the hearing loss back to this level here. And so you can see that with these drugs, we can actually reduce the amount of hearing loss, which is a consequence of noise exposure. And you can also see that the number of hair cells which are injured by the noise exposure, we're seeing about 35% of the cells are missing. But after the treating with these drugs, we can reduce the level of injury. So there are these sorts of opportunities in drugs which are now people are looking at are showing the ability to actually recover the sensory cell loss after things like noise exposure. And this actually works also very well to protect the ear against age-related hearing loss, and also some of the aminoglycosides and ototoxic drugs. But there are really a lot of challenges to be able to translate this research into clinical practice. Number one is that the sort of animal models, because all of this work is in animals, and what animal models do we use and consider those. The other thing which I hear is that it's a real difficulty to understand how these processes actually, whether they occur in humans. All of the work we've got is on animals, and understanding how these disease processes actually occur in the human inner ear is something which we don't have a lot of information about. And the other area which I want to talk to you, would be interested in your comments, is how to actually deliver these drugs into the inner ear. So how can you deliver these compounds, because there's this blood-labyrinth barrier, so putting these drugs in systemically, there's a problem in being able to get them either to the right concentration, or there is also there are side effects from some of these drugs. So try and get those into the inner ear means we've got to find some new ways to do that. So just now I'd like to just move on and ask just some questions about how it is that we can study some of these processes inside the inner ear, and particularly being able to look at some of these in humans. Because the only way we've got of being able to study disease processes really in animals as well is to take the tissue out. We can only, the cochlear is very small, it's encased in bone, all the tissue sits inside the bony labyrinth. So to actually be able to study that, you've got to open it up to look inside. So we don't have apart from some physiological measures, we don't have a lot of ways of being able to study disease processes and then also to be able to do that in humans. There's no way we can take biopsies and so forth in humans. So we've been looking at ways to use MR imaging to study some of the disease processes in the inner ear. And this is an MRI of a guinea pig cochlear taken with a 12T system. This is in Oregon, and that's the histology of the cochlear. And you can see here the auditory nerve, and you can see the compartments here, which are similar. And you can see here is the the areas of the eighth nerve. So the resolution with this particular MR is very very good. But it gives us an opportunity perhaps to study some of the disease processes, and I'd like to talk a little bit about this, which is some of the work which we've been doing here. So what first set of studies which we've done is to think about, can we study more about the vascular permeability and the blood-brain barrier and how that might be being altered during with disease. And we've taken an animal model which we use, which is a labyrinthitis. So this is where we introduce a bacterial lipopolysaccharide into the middle ear, and it gets taken up into the inner ear through a very small window called the round window, just a very, very small membrane that separates the fluid of the inner ear from the middle ear, and this is taken up and it causes an inflammatory response inside the inner ear. And one of the ideas here is if we use a particular type of MR approach, which is dynamic contrast enhanced MRI, the idea here is if we introduce the contrast agent, which is in this case is gadolinium, into the vasculature, then and we're scanning and imaging the tissue as the gadolinium passes across into the extracellular space. It will then cause a change in the magnetic properties that can be picked up by the MR. So we can actually then measure the concentration of the gadolinium inside the extracellular space. And there are a lot of things that will constrain the rate at which that moves across, one of which is the permeability of the barrier between the vasculature and the inner ear.
Peter, excuse me, what's stopping you from going the other way? Or is it preferentially going to one way or another? Or
so in this case, we're injecting it solely into the vasculature. So we're looking at, and if there was an equilibrium in this, but the concentration is very, very high in this, so the passage is in this direction, but there is in fact a two-way if there's an equilibration. But the modelling enables you, because the passage of this, it disappears very quickly. So you see we'll show you the uptake within the tissue. And this has become quite a standard approach for looking particularly at tumours, and particularly looking at tumours, malignant tumours, because they have a very, very high or poor vascular permeability as a consequence of the very rapid turnover of the tumour. So that's being used as so it's a similar technique which is used in cancer biology. So the idea here is if we're scanning at intervals after the introduction of a bolus of gadolinium, then we can start to look to see whether there's a change or get some idea of the pharmacokinetics of this. So in these studies, what we've done is we create the inflammatory response, and then we introduce the gadolinium at a particular time point intravenously, and then we scan for about 70 or 80 minutes, and we're looking at the rate at which that gadolinium is taken up into the inner ear. And this is the sort of response that you get. So here's in here, these squares here. This is where the cochlear is, and so this is an animal which has been injected with saline. So there's no inflammation in these animals, and you see no change, as you can see in the other images here. There's no change in the response from the cochlear with the MR. With the ones which have had the inflammatory response, you see over a period of 60 minutes after the bolus of gadolinium, you see a very heavy uptake of gadolinium inside the inner ear. And this can be just looking at the signal enhancement, you can see over time that this is the case, that there's an increase in the amount of gadolinium, or the increase in the signal intensity, and this is say 4 days after the introduction of the inflammatory response. And if you then look at the same animals over time, because that's one of the things with this technique, is you can look at the changes in the gadolinium uptake over time, and we can see that animals which are, if we take the values of these, at day 0 we see very, very little uptake. At day 4, after the gadolinium, after the inflammation, we see a very high uptake, and then it recovers. So this is demonstrating here over a period of time, so day 4 through to day 10, that there is a recoverable change in the rate of gadolinium uptake, which suggests that this is indicative of, or we see that as being indicative of a change in the vascular permeability. And not to go into detail on this, but using some pharmacokinetic modelling, which was this 2-compartment model, which was by Tofts, is to actually then calculate, once you've calculated the amount of gadolinium inside the cochlea, so that's taken by assessing it against some standards. We can see that the permeability constant, if you like, of the with inflammation increases by about 6-fold. And so in the same animals, we're seeing that then it recovers. So this appears to be then some sort of way in which we can assess the permeability of the vasculature following an inflammatory response. We've then gone on to say, well, does that relate then to other indices of inflammation? For example, the uptake of macrophages, the invasion of macrophages into the inner ear with the inflammation. And one way to look at that is to inject small particles of iron oxide into the vasculature, and that's taken up by monocytes and macrophages, which when they then move into the inner ear as a consequence of the inflammation will reduce the T2 signal inside the cochlea, and that's an indication of the presence of these cells, and we can use some histological evaluation to check that as well. And so the same sort of experiment here, where we're sensitizing the animal to the LPS, doing the injection into the middle ear, and then straight away injecting these small particles, these small iron oxide particles, and then scanning the same animals at periods of time up to about 7 days after the inflammatory response. And what you see in this case, the same sort of thing, is that if we look at, say, day 2 in the same animal, look at day 2 after the exposure, you see this enormous quenching of the T2 response from the inner ear. So this is the response prior to the introduction of the LPS, and here's the response up to 2 days afterwards, and right through up to about day 7. And there's some quantification that you can do of this to look at the changes. And what it seems as if this is, we think, is indicative of an invasion of the macrophages into the inner ear as a consequence of the inflammatory response, and some degree of recovery after that. And you can pick up, it's probably not very clear here, but the blue here is actually the presence of the iron oxide inside the cochlea. One of the problems we've had with this is to be able to prove that this is actually inside macrophages. We've used a number of macrophage markers, but unfortunately the cochlea also has its own macrophage population, so it's producing, the staining is at this stage is a bit equivocal, but we've just got some ideas about how to discriminate that. But it does, at least in our that the particles are getting in. Whether they're coming in, we believe they're coming in cells, but that's still got to be sorted out. And just as a small study which we've been working with some people in Melbourne, we're interested in the cochlear implant produces, when they insert the cochlear implant, it causes an inflammatory response. And so in animals, if you put the cochlear implant in, it causes the inflammation, which is what you see. This very, very large dark area here is once again using these iron oxide particles. you can see this very large area here is indicative of an inflammatory response inside the cochlea after implantation. And if you use dexamethasone as a steroid, dexamethasone, in some of these animals, it seems as if we can actually reduce the amount of that inflammatory response. And so this is is sort of indicative of that in a way as a non-invasive technique. So with this, we've started to do some human translation studies, and we can measure the cochlea. This is the inner ear in humans. There's the auditory nerve, there's the vestibular organs there, the cochlea you can see here. Slightly better on higher power is the cochlea here. Here's the auditory nerve and the vestibular nerve, the vestibule and some of the semicircular canals. And we've started looking at these in a particular condition called Meniere's disease, where we believe that there are defects in the blood-brain barrier which occurs associated with this disease. And we can see exactly the same sorts of things. We're not using the iron oxide particles in this, but just looking at the uptake of gadolinium. And if we look, in this case, this is effectively in a control, you see that the hot spots here, this is the cochlear nerve and the vestibular system. The blood-brain barrier is much tighter, so it takes about 4 hours for the concentration of gadolinium to increase inside the inner ear, but you can see an increase here over that 4-hour period. And what's interesting, if you look at patients who have got Meniere's disease patients, it seems as if the patients who have Meniere's disease in one ear and a normal ear, there is an apparent, these are very, very small numbers at this stage, but there is an apparent enhancement of the signal which comes from the ear which is actually affected, which suggests that in these patients there is a change in the blood-labyrinth barrier associated with these particular conditions. So we're quite hopeful that this is a technique which we might be able to really apply to look at inflammatory responses and so forth in blood-labyrinth barrier defects in human disease patients. And this is just showing you once again, this is a nice one in the particular Meniere's patient here. And what you see is, here's the cochlea here, here's the vestibule, and this is in a control. And when you look at a Meniere's patient, you see a substantial increase in the amount of gadolinium which is taken up in the Meniere's patient compared with the control, which suggests, the only suggestion we've got is that this means that the blood-labyrinth barrier there's a defect in that barrier associated with this particular condition. And we've done some modelling to try and look at the permeability constant, and so far with about 7 patients we're seeing a significant difference in the permeability constant in these patients. So just to sort of come towards the end really, is that what I just sort of tried to show was that there are a lot of different ways that we're looking at the inner ear, but one of the things with the use of the MR is that it does give us an opportunity to look at potentially reversible changes in some of the vasculature at the moment, anyway, looking at inflammation. So we've got 2 markers of inflammation which we can look at in the inner ear. And both of this, whether it be through the detection of macrophage uptake or through the permeability of the... So it does suggest that it's quite feasible for us to use MR imaging to qualitatively and hopefully quantitatively investigate disease processes inside the cochlea. So we're quite excited about where this is sort of potentially heading. But there are some issues which I'd just like to spend maybe just the rest of this seminar just talking about. So the first thing is that it's all fine. We've got some nice images and we've got some nice processes and we're getting some consistency. We've had some real problems in being able to get consistent information from the scanner. We've been a lot of difficulty in being able to work with, obviously with very, very small samples, very, very small amount of tissue, so the size of the cochlea is very small, is to be able to get very, very accurate measures of the gadolinium concentration. And we've played around a lot. When I say we, Jérôme Plumard and Beau Pontré, who are with us, and Beau in Camry, has done a lot of work trying to improve the sequences with the 3T and the 4.7T to improve the measurement of gadolinium inside the cochlea. But we really need to think about ways to improve or to validate our modelling approaches, particularly the assessment of the vascular permeability and the modelling of that. And we'd be really interested if there were ideas that people might have as to, and perhaps some way we could work together to look at improving our modelling approaches. One of the things that we have a problem with is that the signals are there's very, very small signals which we're getting because of the size of the tissue, and trying to get coils which would be much closer to the inner ear and being able to pick up and improve our signal to noise ratio. So we were delighted. We had a fourth-year project, Alec Wang engineering project, who worked with us this year with Paul Nielsen and Beau Pontre and Jerome Prumar, and has made a lot of progress in just making a very small coil, a 1.2 centimetre coil, which we hopefully would be able to use with, obviously there's a lot further to go, but to generate small coils that we can get right up close to the inner ear, a surface coil. But we've even thought about the idea of getting one right down the canal and being able to get very, very close to the inner ear to improve the signal. And the guys that we work with in Portland who do a lot of this work, they have this fanciful belief of actually being able to thread a coil inside the cochlear and study the fluid fluxes, but I don't know about that one. But anyway, a bit of a challenge for everybody. So we want to think also, look at other measures of inner ear function, so not just relying upon the measures we've done so far. And for example, the idea of using manganese, which is taken up by calcium channels with activity, is being able to study using manganese to be able to look at neuronal activity. So start thinking about ways that we might be able to use these imaging techniques to study much more of the function. And finally, the other thing which we really need to think about is that if we're going to go, once we've got all this image, we've got all these ideas about the ways to, and some of the pharmacological approaches, and even with the stem cells and so forth, we have to be able to get them into the inner ear. And because of the blood-labyrinth barrier, getting drugs across the barrier into the inner ear in the right sort of concentrations is very, very problematic. And so one of the ways which and also because of the possibility of side effects, off-target effects with the drugs. And one of the ways that has really been a thruster, I suppose, nearly all the research that's been going on is the idea of using this very, very small window, the round window. It's a small membrane, actually a very interesting membrane. It's shaped like a Pringles chip. And I didn't understand this, but you guys must understand why Pringle chips are the shape that Pringle chips are. And it's the membrane, the round window is shaped exactly like a Pringle chip. And what it is, apparently, is that it is very resistant then to pressure. And so the middle ear pressure changes. So the round window itself is, it's not there to allow drugs and things to go into the middle ear, into the inner ear. It's there as a relief. So the stapes is the one that pushes from the final bone of the ossicular chain, transfers the energy from the tympanic membrane, from the eardrum, but it needs to have some sort of relief for the transfer of pressure. So the round window offers that and moves in unison with the stapes. And in order for that to function, and with changing middle ear pressure, this membrane has a structure that apparently with changes in pressure it doesn't change its shape and doesn't change its stiffness. I don't know, you guys must know that, but it sounded pretty cool to me. But the thing is that this little window offers a window actually into the fluid system. And so the idea of trying to use that as a way of getting drugs into the inner ear has become sort of very, very popular. But one of the problems is that there's a finite size of compound and those charges that will get across. And also, how do you maintain a concentration? So keeping the concentration at a level consistently on the outside that you're getting the right amount inside has been problematic. And so people have been looking, there's one group which has now generated a little micro punch, so going in and producing micro holes, using carbon filaments to produce little holes in the membrane. And that's a bit of an issue because you've then got to have them seal up, or you're going to get infections and so forth in there. Some of the other ideas, and we're working at this at the moment, is to use nanoparticles. So we've got work with a group in Melbourne using nanoparticles which we're loading with the particular drugs, and then we're using instead of using the round window, making a small hole in the bone and introducing those to have slow release of the drugs when we put in things like cochlear implants. But there's also some ways where these particles will actually get across the membrane and into the inner ear as well. And so, and the final thing is that the idea of using cell-based therapeutics. So here is Living Cell Technologies, which is in Auckland here. They use subarachnoid cells to produce, that you can put into sort of albumin gels. And these then can be put into, then some work been going on inserting these into the inner ear to actually slowly release particularly neurotrophic factors. And one of the other ideas is, can we perhaps use approaches to temporarily open up the blood-brain barrier when you're putting the drugs in, to see whether you could increase. So for example, some sort of sonication approach with microbubbles or something close to the inner ear to open up those vessels and allow a greater increase in the pharmacological agents you want to put in. So I'd really like to think that there might be ways in which we could all think of maybe some projects to look at some of these things. We would really love to have some input from engineering into be able to do some of these things, particularly some of the areas around the modelling and and looking at local cochlear drug delivery, because it is really an impediment to getting these drugs into the inner ear. And so look, I'll stop there and just acknowledge a lot of people who have worked with us on some of these projects, and a lot more, and some of the funders that we've had. So, and we're really keen to any questions or comments or things.
So thank you, Peter, for a fascinating talk. So questions, Jeff?
So I'm thinking about the different ways to image the inner ear. Does anyone do ultrasound imaging of that region? It seems like you could make a transducer, right? A specialized transducer that fits in that canal.
Yeah, that's an interesting idea. No, I don't think of anybody that's done any imaging with ultrasound. No. Okay, that's project number one. But I mean, yeah, I mean that's interesting too. I don't know what the resolution would be and things in that. But I suppose if it's a question of getting your probe small enough and being able to focus.
The sound waves wouldn't be a problem.
I don't know. I was
yeah.
That maybe
well, I mean that side of it. Yeah. Well, not in terms of not really. I mean the only thing is that the vestibular system actually responds to ultrasound, but you'd be way outside the frequencies.
Very high frequencies.
Very high frequencies. Yeah. That might be an interesting way to look at it. Yeah.
So, how much room do you have? You know, you've got the round window, and then you've got to get through the middle ear. How much space is there to get instruments?
Yeah. So in the human, the distance between the eardrum and the round window is about 6 millimeters, and then the space itself would be about twice the size, maybe 1.5 times the size of that. So it's quite small, but there is quite a lot of. There is a reasonable amount of room. I mean, people put in wicks and various and use instruments to do the surgery in there, and it's actually accessible as well. So you can get into. I mean, surgically it's easy to get in. You would obviously try to get to something which would be more of an office procedure, but surgically you can get into there and put things and manipulate the round window quite easily. Yeah.
So then to get drugs into that space, I mean, it's kind of awkward, I suppose, and you've got to break the eardrum or at least pierce it.
You can, or surgically that you can. It has a ligament on it, so you can just dissect the ligament and just pull the eardrum back, or make a hole in the eardrum and go in. So at the moment, most of the delivery systems have been either using small pumps or using gels like pluronic gel and so forth to cause and have a slow release. But the problem then is that the window itself, under a lot of disease conditions, also thickens and changes, so you really have no idea what drug concentrations are actually getting in. So being able to actually access it and deliver a known concentration is really what we need to be able to do.
So why isn't, or why aren't drugs injected then, where you can have, measure the quantity?
Well, mainly because the membrane itself is really so small, and the fluid inside the inner ear is you've got to be very careful about putting anything in under pressure. So diffusion is the better way to deliver things. But as long as there was no pressure, but if you could get, and there's enough compliance in the inner ear to some extent to actually take up a bit of volume. But you could actually just So it's, and the membrane itself, I think, if you put in something too large, of course it'll split, and then it's the question of the repair of that membrane and then the leakage of the fluid out afterwards. So it does present quite a problem to get something which will actually be made.
But you can get very, very thin, I guess, pipettes.
Yeah, I think that I'm just trying to remember the dimensions. I should have had that. It's about, I think it's about 600. No, it must be close to. It must be about 800 microns or so. So it's not that large. So yeah.
How much impact does that have on hearing function when you actually got something in there?
You smash the Pringle, or actually in the middle ear.
Say a pluronic gel, which you're actually applying some
Well,
fluid into that.
yeah, I mean, mainly if you've covered the round window, then and it's no longer actually able to work in its normal function, then you will have some conductive loss. But the thing is that that's going to be temporary. So it's really and as long as if you look at the cavity, the eardrum and the round window sits on one side, so you've actually got it's quite a nice, it's reasonably accessible. There is a problem is that it's sort of recessed into a little cavity, it has a little recess which makes it a bit more difficult to get to. But it seems to me that some idea, you want something which you could puncture it, but actually leave that puncture in place. So then you can deliver compounds and be sure of what compounds you're actually, or the concentration of the compounds that you're putting in.
You're experimentally induced damage to the blood
Barrier. Labyrinth.
something or other lymph
Labyrinth, yeah.
labyrinth barrier. That enhances the inflow, if you like, or the in-crawling of macrophages. Is that right?
No, I don't. We're not sort of thinking of it. I mean, it may do. It may. I mean, opening it up may well change that. The timing relationship is interesting, but we haven't really sort of investigated that much further. But we're just sort of assuming that it's actually indicative of the presence of an inflammatory response. So it's an attraction for those cells into the inner ear as a consequence of the inflammatory response. The actual triggers and the relationship between the signals which must be being sent from the cochlea to the vasculature to actually attract those is of interest as well. But that's the way we're sort of thinking of it more.
And you were imagining that in one of your protocols they're ingesting the iron particles.
Yes, yeah.
How did they then get back out again?
Well, I don't think. I mean, unless the particles are being removed, we haven't done the quantification yet. So the particles may be being removed by there is a sort of phagocytic process within the inner ear, which then takes material to the inner lymphatic sac and so forth, which then removes it. So we're sort of assuming that there might well be those sorts of processes that take. There is a garbage disposal. There's a waste disposal unit in the inner ear.
Must be saving the lymphatics, which actually act as the drainage for the entire system. There's got to be some kind of clearance on the compartment, isn't there? I think?
Yeah, but it's actually. I mean, there is a vascular clearance and there's a small sort of lymphatic clearance, but it's mainly through this other duct, which takes it, which removes that material. I mean, that's actually it's not clear. It's what's surprising is how tight the barrier is. So the inner ear actually is very, very heavily protected. And so it does, and conditions like Meniere's disease and so forth are thought to be, one of the theories there is you've actually set up some sort of autoimmune response where something gets through into the cochlea, generates an inflammatory response, and then it just starts to become amplified because it's so well contained.
And presumably there's some very good survival reasons why this could be the case.
Yeah, I mean, in the ear, I think that's absolutely it's the most critical of your senses. If you both balance and hearing for survival, because it gives you it's a.
Or plus access also of fiction through that part.
That's and that side as well. Yeah.
So opening up that barrier for drug delivery could come with the other side effects, which could not be so pleasant.
Yeah. So that's and one you might be able to. I mean, there's a lot of work which is going on trying to do this in the brain, where there's a number of compounds and the idea of microbubbles and various things to open up the blood-brain barrier. And it's just transient, so you're looking at just transient changes. So the possibility of doing something similar, and if you had something like an ultrasonic way of being able to, which could be quite focused, maybe you could actually increase. One of the interesting ideas is that maybe even using similar sorts of approaches to actually open up the round window during, so you could actually change the permeability of it using maybe ultrasound or something, and then allow the drugs get a better concentration of drug in. But it's a problem. Yeah, it could potentially have additional side effects. But
so could drugs be loaded into macrophages, and since they already make their way through membranes and everything?
yeah, I suppose the problem with that is that you don't really necessarily want them. I mean, they're indicative of some, but I suppose if they're getting in, I think the interesting thing is the idea of using other either other cells or nanoparticles to be able to get the compounds actually get them in as there's one group which has used these carbon cages, and they've then attached an antibody to the carbon cage which ties into the neurons, and those then have drugs or whatever inside them, so then they circulate, tie into the neurons, and then they start releasing the neurotrophins. So there's a lot of those sorts of, looking at ways that you can guide the drugs to the right place within the inner ear is part of the problem too.
Oh, I just wanted to know, so the nasal cavity is somehow connected to your ear as well, or is that through the middle ear?
It's through the middle ear, so that's the eustachian tube. So the back of your nasopharynx, there's a tube that goes up that connects the middle ear, and that maintains the aeration of the middle ear and pressure across the eardrum. Yeah.
And so could that be a potential way to get?
Well, the only thing you can achieve is to get drugs or compounds actually into the middle ear, and then it's a question of to get those.
So, yeah,
You still don't have access. Yeah, I mean the compounds that we've been working with, we can get them across systemically. So we've actually been able to measure the concentration inside the inner ear using LCMS and taking samples and measuring the actual uptake of these compounds. But when we've tried to go into to get patents on some of these things, because they're agonists, which is not the sort of thing that you would normally be putting in, but they're agonists of adenosine receptors, for example, which then have effects on cardiovascular effects, that the pharmaceutical companies just run away from it. So if we can we can actually have local drug delivery, then those problems actually would go away. And we've got ways now of being able to amplify the response of the drugs that we've been using, and so we'd really like to try and find ways to get them specifically into the cochlea.
Peter, just, I'm sure you told me this before, but the ADAP, the adenosine receptor agonist, was it that you were saying that you were going to explain? That acts via the outer hair cells, or?
Well, we don't really know. The ADAC is so the cochlea has all these purinergic receptors, so ATP and adenosine receptors, and ADAC So the A1 receptor is very much with the supporting cells. And we think of the supporting cells inside the inner ear as actually being they're a bit like glial cells, so they protect the hair cells. And so what you're doing is modifying their response, and they seem to be the ones that are generating a lot of the free radicals, and or partly associated with that. So the adenosine receptor seems to mute, certainly the A1 receptor mutes that oxidative response. Yeah. But then there's the A2 receptor, which may in fact be may have a problem. Yeah. So.
How consistent is the blood supply and lymph supply to the inner ear across different individuals in the population?
It's a good question actually, because it is quite variable. The vascular supply, it's a terminal vascular supply, so there's only one vessel that goes into the inner ear, and it comes off the basal artery, but some people have much better vascular supply, and there's actually or have numerous vessels going in. Others don't. So there is quite a lot of variability in the vascular bed. Yeah, it's a very, very metabolic structure. It has a very big blood supply relative to its size, because of trying to mainly maintaining these fluids, because they're pumping ions, and particularly maintaining this very high positive voltage inside the ear, which is essential to driving the whole process. So it's actually very metabolic and has a very, very big blood supply relatively. Yeah. So if there are, what I tried to do is just to sort of give you a bit of an overview, so I'm sorry if it's sort of a bit broad, but it's just that I think there are some really interesting issues and problems. The MR is really giving us quite an interesting tool to study it, and it's been pushing the boundaries a bit. But we would really love to sort of try and engage more with engineering to try and solve some of the problems we've got. So we've had a wonderful experience with Alec and doing with the fourth year project. So we'd be delighted to possibly keep doing those as well, and offering projects for students. So anything you might think of, we'd be delighted to talk to you. Thanks very much.